for the brief introduction and welcome. Now we'll start with the invited lecture on the use of molecular genetics in animal breeding. First though, a few words about Dr. Lutz Bunker. So Dr. Bunker is a leader of the Growth Genetics Section, Animal and Veterinary Science Research Group, Animal Breeding and Genomics team at the Scottish Agricultural College, Roslin Institute Building, at Easterbuch, which is a bit outside of Edinburgh. So he's German, and he got his PhD in 1979 from the Academy of Agricultural Sciences, Dummersdorf, Rostock, Germany, with summa cum laude. As for his research interests, he worked before joining the Scottish Agricultural College in 2002, and his work then focused on experimental population genetics with mice, and 15 years at the Research Center for Animal Production in Germany, and about 10 years at the University of Edinburgh, working with Professor William Hill, and using mice as a genetic model for humans and farm animals. So he had initiated one of the longest selection experiments in mice of over 130 generations, which produced the heaviest mouse line in the world. Hopefully we'll see a photo of that in his presentation. And helped to develop also the smallest existing mouse line just by simply breeding them divergently for size. So his interest focused on studies into the genetics of obesity in mice, on mapping and fine marking quantitative trait loci for quantitative traits including growth, body composition and metabolic rate in various long-term selected mouse lines, and on candidate gene approaches to validate or disprove the involvement of candidate genes in certain metabolic pathways. He's also been working on marker-assisted introgression studies and selection on the single gene level, and he developed techniques for semen sexing in pigs. He studied genetic polymorphism for boar-taint in the porcine myostatin gene, and he worked on identifying and validating QTLs and genes that affect muscle growth and other production traits in pigs. Additionally, he worked on evaluating new technologies of potential use in pig breeding, including the development of business proposals. So, since he started to work for SAC, his research interests remained in the field of quantitative genetics and the use of molecular genetics and their application in farm animal breeding. But his interests were also extended to the use of imaging technologies, mainly computer tomography and video image analysis in animal breeding and research and into studies of milk quality. At SAC, he's leading a section of 3 scientists, 4 technicians and 5 PhD students, working mainly on sheep genetics. The computer tomography unit and the experimental sheep unit are part of this section as well. And since being at SAC, he has been project leader or senior researcher for almost 7 years of several DEFRA, CIRAD or LINK-funded projects, mainly related to the genetics of sheep, disease resistance, growth, body composition, meat quality and meat eating quality using computerized tomography and video image analysis systems in abattoirs. He has over 140 publications in refereed journals starting from 1978 and up to 2012, and he regularly reviews scientific papers for Meat Science, Small Ruminant Research and other journals. And Dr. Lutz Bunker had taken an active and useful role as consultant in the research project on the use of molecular and quantitative genetics for the improvement of the Hίου sheep breed. So I'd like to welcome Dr. Bunker for his talk.
Thank you very much, Georgia, for this nice introduction. Nice and comprehensive. I must be very old if I have done all that, what she said. So, yes, and I mean, it was, many thanks also for giving me the chance to speak to you, which is not a simple task, because usually, I mean, I do 90–95% research and only 5% roughly teaching. But on this subject I teach usually 4 lectures and each lecture has like 2.5 hours. So my task on Wednesday and was not easy because, you know, one thing, you come from Scotland, we have in the moment 6 to 8 centigrades. And coming over here and then giving up the whole Wednesday to condense these 10 hours into 1 hour, keeping in mind that not everybody is a geneticist in here. So I tried actually, you know, to condense and simplify the whole story. I hope I was successful. If something is unclear, just interrupt me and ask directly on the spot. That would be probably the easiest. But my time is limited, so 50 minutes, maximum an hour, I think. Okay, you had enough time to read the title. This is a kind of emails you get nowadays. It's just, you know, just one example. Every day you get these kind of emails, job offers. Now go essentially professor of animal breeding. And the thing is, the field of professorship has specified on the use of genomic information in animal breeding applied in this case to pig breeding. But this is the kind of job offers you get nowadays. So it's always a connection between quantitative genetics, so the classical animal breeding, and molecular genetics. So, you know, I could have shown many of those. The problem for today is essentially there is so much information out there, and the available time is so limited that it might be too much, might be painful. So let's see how we go on here. So we do the 1 hour service here. And so I try to simplify the genetics part, and you know, everybody knows we have, sorry, that was too fast. We have one chromosome is from the ram, and one chromosome is from the Eve, mom and dad, so to speak. And chromosomes come in pairs, and sheep it is 26 pairs, and plus the X and the Y chromosome. And on these chromosomes you find them all the genes. So genes are spaced along the chromosomes, and then these genes produce essentially the proteins, and they have the blueprint for the proteins, hormones, enzymes, etc. And that means they regulate they are involved in very many metabolic pathways. So if you take that picture, you can actually see that they produce finally the phenotypes. If you take this as an example, like the hind leg muscularity, so this is all determined somehow in the genes. And there are many more metabolic pathways if you also bring in mutations, which are just, so here's a mutation happened on both chromosomes, so it's homozygous, this animal. Here it's a heterozygote on this gene, heterozygote on this gene. So this gives actually the opening for very many more metabolic pathways or disturbances in certain pathways. It is then a little bit more complicated than just a simple line. So essentially you have, let's say, this double helix here. So you have your copy from the ram, copy from the Hίου, and the chromosomes are strings of paired bases, so the double helix here. And so you know that the bases A and T are paired, and the G and C are paired, and so on. So, and there is a base change, which is like a mutation every 500 bases, and we are talking about single nucleotide polymorphisms, the so-called SNPs. And so if you look at SNPs, then here we have 5 different sheep. There's a base change every 500 bases, as I said, and here you see essentially like, here you see a difference, all of them are G here, but this sheep is A, and so on. So you have actually several million possibilities for SNPs. And there are, in addition to make it more complicated, there are also inversions, deletions, gene duplications, and so on. So, and sometimes such a little SNP doesn't matter at all, because you don't see a difference in the phenotype. Sometimes it does. If you look at this is a mouse. There's only one SNP one nucleotide difference between this mouse on the left and this mouse on the right. This is affecting the so-called lip mutation in mice, and there's only one nucleotide substitution, and that means it leads also to a change in the amino acid sequence. And what you have is these mice don't produce any growth hormone, because the lip mutation affects the growth hormone releasing receptor, and that means finally these mice don't have any growth hormone, but they are viable, they produce, these have a litter size of 10, they have a litter size of 2. So just to the size of the task essentially it is quite large. So the predicted genome size in sheep is, for example, 3,400 in this case centimorgan. But we come to that one. I hope you appreciate what I did here. I tried actually to manipulate this typewriter and took all the keys away, apart from the 4, which I told you before, so that we have still the building blocks of the DNA we have here. So it's like you type a very long text just with these 4 characters. And the genetic code is written just using these 4 different characters, but it is 3 billion characters long. Or you can, if you think in computer terms, 3 gigabyte, which is nothing anymore for a hard disk size, but for the RAM maybe. Regardless, I typed this text just to make this little example. So if you just imagine the size of the genome of an animal, which you have in every cell of that animal, then just think about that one. So this is like 3 gigabase base pairs here, and roughly maybe 30,000 genes. And if you think about that on one normal page A4, you get roughly 57 characters per line, 36 lines, that's what I did here. This is roughly 2,000 characters per page. This gives you like, and if you think about it, you put 1,000 pages in a book, on the airport, these kind of things for traveling. Then you have like 2 million characters per book, and that would give you 1,500 books. If you think about one book 5 centimeters, that's a big one. So then you have like 75 meters bookshelf space. That is in every cell. Just boring text, I mean, who wants to read that one? But we are nowadays very keen to sequence, of course, the whole genome. And then you have your 75 meters on the bookshelf here. And then think about that one. This is one snip in the whole lot of these books, and it can matter or it can't matter. And then if you think further, that if you are talking now about the use of SNP chips, a 50K SNP chip gives you essentially one SNP on all, roughly 30 pages in these books. So we are very proud to have now a 50K SNP chip or a 700,000, the high density SNP chip, and this gives you then all 3 pages, but there are still lots of opportunities for SNPs which might matter, which you won't discover. That's just the task. And then if I just take you back by 20 years, 20 years is roughly the time we have now molecular genetics in the animal breeding field, if you so want. That was the same time as we introduced the World Wide Web. Who could think nowadays of not using the internet? We put our books away and just rely on Google, don't we? Then we have, 20 years ago, Motorola released the first hand-sized digital mobile phone, which is probably that size. So think about the one thing what you have in your pocket. And 20 years ago, DNA fingerprinting was beginning to be used commercially. This is a technique that matches organic materials to the individual they came from. So fingerprinting became a standard, and then it was the beginning of the Human Genome Project. So just to remind you, the completion of the Human Genome Project was roughly in 2002. I mean, it obviously never ended really, but you had to say, okay, we have now sequenced the whole genome of a human being. It took at this time 10 years of research and an amazing $2 billion. And And the Human Genome Project finally reported that 99% of the human genome had been sequenced. So this is all the 75 books they went through and could actually read that one, all in the boring code. But we have, I just show a few here. Meanwhile, we have also the genome from the honeybee, we have also the gorilla, we have also the chimpanzee, we have the bovine mouse genome project, and all these followed later on. So genome projects are scientific endeavors that ultimately aim to determine the complete genome sequence of an organism, of an animal, a plant, a fungus, a bacteria, a virus, and what have you. And to annotate protein coding genes, to recognize actually what are the genes in there, and other important genome encoded features. So the genome sequence of an organism includes the collective DNA sequences for each chromosome in the organism. So for a bacterium, it's just a sequence of one chromosome. For the humans, for example, 22 pairs plus the sex chromosomes. Okay, and what can you do with that one? You take just a simple, a group of healthy people, and here you have a group of unhealthy people. You do the whole sequence, and then you try to identify the genetic factors which are involved in disease, but could in principle obtain the complete genetic sequence of several individuals, some with the disease and some without, and then search for the difference between the two sets of genomes. And because we are talking sheep or farm animals in general, of course you can do the same if you can afford it. And nowadays I was told that you get actually a tenfold coverage of the whole genome. Roughly it will turn to $1,000 per genome. So it gets affordable. Just think about it. That is, in the moment it is still expensive, and some people think it's infeasible because of the cost, and therefore we did some things like the HAPMAP project. So because this was supposedly a shortcut, that you used some sort of snip chips for before you go to the full sequence. So there was an international, or still exists, an international HAPMAP project led by the Australians, James Caius, and the organization whose goal it was to develop a haplotype map of the bovine genome. And the haplotype, just short definition, is a set of single nucleotide polymorphisms on a single chromatid that are statistically associated. I don't want to go into more detail what we are talking about haplotypes. Okay, and then we have the snip chip, just a picture here. You have very short ends of DNA sequence are actually stuck to your chip, like showing here. And then you can essentially, only DNA which has the complementary sequence will actually hybridize with that one, or the rest you just wash off after that. And you do that at the same time for 50,000 SNPs. And don't think that is easy. It is essentially the design already determines the outcome of the whole approach, because some people say that the design of these SNP chips should actually be in such a way that you have more SNPs, not just take randomly known SNPs, but take those which are very close to already identified QTL. And I'm stressing that one because this doesn't mean, this all the technology does not mean that we swap tomorrow only to this and to sequencing. There is still value in using microsatellites and using actually more simplified SNP tests. And so there is still room for for many years of research, I think, before we just use this kind of technology. So here we can essentially then identify carriers of certain SNPs and the non-carriers. And what is genomics? Just a quick one. So I just focus on that one, just considering the time. What individual genes do, how the activity of genes is regulated, how genes are influenced by those around them and their environment. These are the most important things that we want to get out of that one. We had last week Jerry Taylor from the US in Edinburgh, and he was saying, you know, everyone was very full of enthusiasm about the use of SNP chips. Now people say, but it's also very important, you know, not just using them for estimation of genome-wide breeding values. It's still very important to identify the genes which are underneath. Because finally, without the knowledge of those, we can't design our SNP chips very well, and we still want essentially to get to the bottom of all that. So it's not just, you know, the high throughput genotyping. I think I can skip that and tell you a lovely story about a boy. He is called QG, so it's quantitative genetics, and the girl is MG, molecular genetics. And I was in 2001, my goodness, it is already 11 years, I was in Iowa. And there was a conference, they were talking all the time about the vetting of quantitative genetics of animal breeding and molecular genetics. So I tried to tell you the story why. But who are they and what did they do before? So I mean, QG is obviously a very old guy, and as man domesticated, they selected immediately, and QG might be over 10,000 years old if we so want. How powerful was and is breeding? And this is an example here for the cattle world. So essentially it went up from, let's say, 6,000 liters, it doubled more or less. And of this one is at least half of this one is genetic change, because over these years you can imagine we improved also the environment quite a lot, but half of that one is genetic change. Down here you have a plant example. I don't want to go into details of that one. This is a nice one because it summarizes. So Van der Steen summarized actually the selection response or success of breeding for quite a few species. So he was saying that pigs weaned per sow and year jumped from 14 to 21, so 50% change. The lean percentage of the pig world went up by 37%. The kilogram lean of meat per ton of feed went up by 100%. So we have also improved food efficiency quite a lot. Broiler chickens, days to 2 kilogram because that's the slaughter age. That's the slaughter age. Sorry. So that is reduced by 60%. Food conversion ratio 43%. Eggs per year went up by 30%. Eggs per ton of feed 80%. And milk cow lactation 67%. So quite impressive, I think. And you can also, of course, see when you look at the variation which we have produced over the centuries in the pig world. I mean, you know, the purpose of the pig was at certain times different. We needed them for large production, so we, you know, this was our aim to produce. And until we had them, yeah, more in the recent years we had them this kind of type. Okay, and just to think about that one, I only want to point out here that one measure this in generations, it's probably 4,500 generations since we have domesticated the pig. And here I have also to speak about mice, sorry. I spent so many years in my life with mice. And here is the experiment which we did in Edinburgh, and you have essentially produced the high body weight mouse and the low body weight mouse, and we have a difference of 1 to 3 produced. And as I arrived in '94 in Edinburgh, it was also the time as actually the molecular genetics has more and more developed, and we could now ask different questions than just think about it. This one was probably 72, something like that, 74. And at those days, the aims of such selection experiments were totally different. Now, as I came over in '94, we were full in the hunt for genes, identifying QTL, identifying the genes which make these guys so heavy and these guys so small. So that is, as Georgia said, that is the biggest mouse in the world. I produced it, I started with that one as a young PhD student. So to see the difference, so same sex, same age, there's no trick, no genetic modification apart from breeding. So it's 1 to 6 you can actually produce in a lifetime in a mouse, because you do 4 generations a year. Of course, if you look in other species, that is maybe not that impressive, but of course they use the genetic variation which was in different parts of the world. one. So you have in chicken you have 1 to 5 to 12, in rabbits you have 1 to 7 to 8, and in dogs you have 1 to 40 to 80. Just to show you these pictures. So that is a layer chicken, and that is a broiler, same age. You have that little one and the big one here, and the chicken broil, and that's of course that's a San Bernard and that's a Chihuahua. So you can obviously keep a Chihuahua in a big mug, in a big coffee mug. So but it's 80 kilogram and 1 kilogram roughly. Okay, so quantitative genetic approach was based on the population genetic parameters for the traits of interest, heritabilities, genetic correlations, and so on. These can be derived by statistical analysis based on phenotypic data and the underlying pedigrees. However, the genetic architecture of the trait itself is a black box. That is still for QG, for the quantitative geneticists. So you have your environmental factors, you have your black box, the animal, and you measure some phenotype at the end, but you don't know what's going on inside of this black box. And so we believe in quantitative genetics still in Fisher's infinitesimal model. And so talking about an infinite number of genes, 30,000, maybe it's close enough to infinity. And all of these genes have very small effects each. So that is a model, and it was quite successful because all the breeding worked so far. But what about the number of genes? What about the effects and frequencies of the genes, the gene-gene interaction, gene by background and environment interaction, and the location of the genes? Quantitative genetics would not actually come up with a solution for that one. Why do we study quantitative traits? In the medicine, most people die from a disease that is quantitative in nature, for which the susceptibility has a strong genetic component. In agriculture, domestication and subsequent selection of crop, plant animals were arguably the most successful enterprise carried out by humans. And in evolution, natural selection works mostly on differences in traits that are quantitative in nature. So the main conclusion at this point is conventional selection works. Never forget what has worked well, but further development needs molecular genetics, and so it's time maybe for wedding between these two. Okay, so what about her? I said already some about her. She's much younger, and we won't understand why we should get married without looking into her history as well. So you have seen all that one, you know, this is quite impressive how much information is in every cell. And regardless if you believe in this or if you believe in this, finally the blueprint of all that is given here in the DNA. And just, I don't want to give you all the timelines again. I just want to remind you that it was 53, and before that we didn't actually know the structure of the DNA. And he came up with the concept of genes actually, and that was Mendel, with his P exponents. And I showed you that, just to remind you, that is all this information. And finally, if we then discover a gene, if we have identified, it was also the mouse which helped us actually before that. Many people found QTLs on chromosome 2, for example, near the myostatin region, but nobody had a clue which gene could be actually underneath. And then suddenly somebody knocked out the myostatin gene in mice. And in the same year you had like 3 or 4 publications of the cattle people, and they identified now, hey, yes, we had all the time, we had our famous QTL peak above this gene, but we didn't know what the candidate is. Now somebody knocked it out in the mouse. Suddenly they know essentially that gene, it was a myostatin gene, which produce these little Schwarzeneggers. So in mice you have a mutation, in dogs you have it, in horses you have it, and mainly you have it in the Belgian Blue and Piemontese and other breeds. But the interesting thing is essentially, once you know the structure of a gene, so you know exactly how many nucleotides are in the promoter region, you know how long the intron is, another exon, you know that this gene, for example, has 3 exons, and you have the introns, you can actually search specifically in this gene for polymorphisms. And that's what some people did, like the Belgians around Michel George. What they discovered was actually that, I hope you see that, it's the same structure of the gene. They found several polymorphisms in here, and some of them obviously you need to knock out the myostatin action to have this double muscling, because it's an inhibiting protein. Muscle growth inhibiting, so if you knock it out, you have actually this very strong development of the muscle system. And I'm just trying to show you how helpful that is. If you know now the gene, you can actually look specifically in this gene for other mutation. And they found 10 different polymorphisms, and some were associated with this polymorphism and some were not. And interestingly is also that some of these polymorphisms in one breed lead to a drastic effect on muscularity, whereas in other breeds they don't. It's a very moderate phenotype that you have. So it's interestingly that it is not just, we call that of course a major gene suddenly, but in some breeds we would actually, the term is not really right, because the effect is quite moderate. But that is just, I hope I was able to show you the big potential which you have once you know actually the exact structure of a gene in here. Another example that was about genetically modification. I visited some people at the Baylor College in Texas. That was as I was working for a pig breeding company. They were interested in genetically modification. Genetically modification has always, especially in the UK, you know, every person is just going away from genetic modification. What a rubbish. I mean, you know, in animal breeding we use genetic modification which are done by nature. Everything what goes in the right direction we select for it. But if the human being does it, then obviously it's all wrong. But I saw there that actually their target was growth disorders in human beings, and you know, if you are a patient, so if you can't produce growth hormone or not sufficient growth hormone, it's actually quite painful process you have to undergo, because the half-life of growth hormone is very short. So it's only 20 minutes, say 20 minutes or something like that. So that means you need actually injections all the time. You need actually to be injected several times a day to have really an effect, to have growth hormone enough in your body that makes you grow. Whereas so these people actually said we go one level higher up, we go to the growth hormone releasing hormone and try to manipulate that. If you have this knowledge of the genes, you can essentially, you know finally also which part of this exon is finally responsible for the attaching site of an enzyme which breaks down your growth hormone or the growth hormone releasing hormone. So if you manipulate actually this bit here, then you can actually destroy the attaching site of that enzyme. So that means you can manipulate the half-life time of a hormone in the system in a quite drastic way. That's what they did. They destroyed the attaching site for that enzyme. The hormone was not so easy to degrade in the body. That means it was longer in the system, longer half-life time. If they have increased, they did that in pigs, they have actually increased the growth of these animals by 30%. Just to give you an example of the potential that you have, if you go now from quantitative genetics into molecular genetics, you know exactly what you're doing. It's almost like an engineering task. Okay, I carry on with that. So you have essentially, you have what are markers, an allele of known position on the chromosome. Definition 2 is a genetic marker, is a known DNA sequence that can be identified by simple assays and which is associated with a certain phenotype. So there are several possibilities to use markers. You have genes, you have known genes can be linked with other genes and serve as markers. Even proteins can be used as markers, allozymes for example. You have then DNA-based markers, the variation microsatellites, and you have these SNPs, which I already mentioned here. So there's quite a lot of markers, and in the moment the SNPs are, of course, because you can put so many of them, you can assay for so many of them at the same time. So why is quantitative genetic a good groom? The improvement of populations by estimation of genetic correlations, heritabilities, and known the pedigree gave already large and huge selection responses in various species. You have seen examples for that one, and that is all built on the infinitesimal model finally. Why is she a good choice for quantitative genetics? Because she can further improve selection response, selection speed, using molecular genetic scores or similar, and it will be a more accurate selection. It can be an early genotyping, because essentially you could, also when you use embryo transfer, you could actually take a cell of the embryo and you can genotype it essentially before you implant it. So you can be very, very fast, you can actually bring down the generation interval, and you can also genotype for traits not expressed in males or in females, take the milk performance. And the cost of genotyping is going down all the time, and you can also genotype for very difficult to measure traits. We have introduced testing stations to measure food intake and all the rest of it. How could you do it outside? Or measure meat quality in a very controlled way. That would be all possible, if you have essentially your genes identified, or if you have very good markers for this. So QTL is a quantitative variable segment on the chromosome that harbors one or a few genes significantly associated with the phenotype. So that is a typical picture you get when you analyze these kind of data. In a QTL study, we did a few with these mice lines. Of course, we started off with QTLs. Then you do fine mapping, and what you then discover is that essentially under one of these curves, there's not only one gene, sometimes it's three, and you were proud to have 11% or whatever of the phenotypic variance is explained by that one QTL. And then you see, like the Matryoshka principle, the Russian dolls, you look then further, you do your fine mapping, you come closer and closer, and then you identify, there's a few genes actually in this region, and every of the genes is finally explaining then only a very small fraction of the variance. So sometimes you get again very close to the infinitesimal model. Okay, that is your... And here QG observes, for example, the jiggle shape variation in sheep. That is done with CT. You can see there's a lot of variation in here. And quantitative genetics would not be able, for example, if we know a gene underneath, so quantitative geneticists would essentially be able only to see that normal distribution here. Whereas if you know the gene, and we are talking about, take myostatin or take another myostatin gene and sheep here, then you essentially identify the homozygote carriers, you identify the heterozygote carriers, and you identify the wild types of that one. So you could essentially use that one. I hope this demonstrates actually how useful it is when you know underlying genes. You can immediately genotype for them, and you can use it in your selection. You can identify these guys, and you say, I take only those. And you fix this mutation, this polymorphism, after a few generations. And then you have homozygote animals. Of course, you need also, we had a big experiment here in Edinburgh where we had a QTL, it was a TM QTL, it was called taxamuslin QTL. What we wanted to avoid is essentially a similar thing like calipush in sheep. Calipush is an extreme expression of the hind leg muscularity, but it's associated with very bad meat quality, very tough meat. So that means every time you are looking at such a gene, you need also to look for possible side effects. Anyhow, how does it work? The principle is actually you have here your quantitative trait, which you don't know, you have not much knowledge about it, but here you have markers. So that is an observed marker alleles, and a genetic marker needs to be highly polymorphic, needs to be abundant, neutral, and codominant. And just to, I don't want to show you very many examples for major genes. They are out there, like borula, myostatin in Texas, or in cattle, and IGF mutations in the pig, for example. But be aware that these are very rare. So this is like how often they occur with the size of the effect. So if the smaller the effect goes, these ones are very rare. So to find actually major genes is a very rare event. And then you have markers which are directly in the gene-assisted markers, if you so want. Then you have marker-assisted selection marker. So you have essentially markers which are sitting outside the gene. And then you can have also two flanking markers, mass markers. And then it's of course a question, the closer you marker to the actual gene is, the more useful it is, because finally recombination can actually break up the linkage between your gene and the marker. And then if that is very far away, then of course the recombination event is going to happen here, and you lose the information content of that one. Just in short, the use of molecular genetic data for selection, the pros are you have inheritability of one, if you so want. It is expressed in both sexes, expressed at an early age, even embryonic, and is useful for difficult to measure traits. And on the cons, it will often not account for enough variants to be used on their own. You have the danger of recombination, and you have the different types of selection, marker-assisted selection, gene-assisted selection, or LD mass, or linkage equilibrium mass, which is more or less useless. So the vetting again, possible selection strategies. You could solely select on marker information. You could construct a molecular genetic score. Then you could do tandem selection on marker score, followed by a normal EBV, blood estimation. And you could construct these kinds of indices, so you have from the normal EBV, and you have that one, the variance which is associated with the markers. I don't want to bother you with that one, I skip that one. This is maybe an interesting one for general, because we have in the UK, and I'm not quite sure about the legislation in this country. If you are talking, you want to do a genome-wide association study, and you need 5,000 animals, you need also to think about, you need to bleed them all. This would be a big task to get from the Home Office the permission to bleed 5,000 animals just for research purposes, if you so want. So you have here one new thing, which is like these kind of special ear tags. If you fix an ear tag to this animal, there's some tissue is harvested automatically in this tube. No typo errors in the field, because if you are doing this in Scotland in the rain and you need to fill in your list, it can happen that your papers are going down in the drain, and regardless, you have the same ID which is printed here. You have actually on the tube. You break that one off. You collect all the tubes. You can send it for genotyping. Here you have the mouth swab, and we are trialing in the moment the nasal swabs, which seem to be even better, because you have a lot of contamination in the mouth, whereas the nasal swab seems to work fine. It seems also that the Home Office is quite friendly to this method. They say it's not very aggressive. You can probably get away without having a personal and a project license. That's very important. You have seen probably these kind of FDA cards where they then take out just a few holes, and you have enough DNA there for, mostly not for SNP genotyping, so for sequencing, but you have if you are after some individual genes, you can use these ones. The blood, of course, then you have the hair follicles. You have Catapult came out with this. This is the nose of a sheep. You do this little prick. Again, the Home Office is undecided. At the moment that you have in mice, you have a little. You take a millimeter out of the ear, and it's enough for getting sufficient DNA. Okay, so what what did we do a while ago? That is a few years ago. We had like, let's say, these microsatellites, and we put them all over the chromosomes. We did our QTL studies. We used our high mice, high body weight mice, low body weight mice, high fat, low fat. And in pigs you did a similar thing. You wanted to actually use extreme, you did experimental crosses. And then you put actually your markers all over the genome. That was like 10 years ago. Then you identify the different carriers of that one. Don't want to go into detail about that one. This is maybe an interesting one. If you haven't seen a PCR ever in your life, think about that one. It's essentially easy. Like you have your parents here, and you see essentially your products walk a different distance in your gel. These people can tell you all about it. But it's also quite interesting. I found it interesting going on a course in Minnesota and did actually some PCR myself, because even when you come from the quantitative genetics side, it is quite interesting to see, also to learn what can all go wrong when you do these kind of things. So coming back to that one, this is obviously the smaller fragment which walks further. So this is Meishan, this is large white. Now you have your F1 animal here, which has obviously both alleles, so it's a heterozygote one. And here you have, this is pictured here, it's a little dirty here, but regardless. You can actually see now all the offspring in the F2 segregate again for that one. You have now, and you can easily select these animals you want out of that picture. So then you do all these kind of things, you end up with your statistical analysis showing this kind of curve. You have here a QTL. And so, and that is an example for the first molecular genetic test which was really of practical use. You know that probably there was a big problem regarding the Stress syndrome in pigs. Many pigs were not able to carry their body weight to the abattoir because they died. And then they identified essentially a gene in the pigs, and they developed an RLFP marker for that one. And this was really a very high uptake of this one in the pig world. And they eliminated essentially this allele, which was called hallucine, from the gas which was used for anesthesia. And this was associated especially with very bad meat quality. So you have here, like, let's say, the lower water binding capacity and PSE meat. This was associated mostly with the gene, and this test was a very valuable one. You see another major gene. So this is the Belgian blue. So that is certainly a homozygote one for the muscling. We have a similar mutation in mice. I did an experiment. So we have a natural mutation in mice where the propeptide is actually, there's a 12 base pair deletion in the propeptide. And I don't think I need to show you that is a wild type. That is a homozygote animal for that one. So you see probably the cross-sectional area. You see also the breast muscle. So it's the myostatin mutation is affecting the whole of the body, whereas some other genes affecting only the hind leg side. So it was actually amazing that this guy who developed these mouse line could actually see through the fur. You know, he saw actually that they must be more muscular. He then selected those and has just reproduced with them. And he derived the so-called compact line, and finally it was found that they are all homozygote for the myostatin mutation. So what we did was essentially, now we were asking, you have now a mutation and you know about it, you have not only markers, you have actually, or you have essentially markers in the gene, you can genotype for the mutation. So what we did was we took it actually and built it into we did a marker-assisted introgression. We put it actually into this high-grow mouse. And you know, the principle of marker-assisted selection is essentially that you have, so this is symbolized here, that you have some good trait which you want actually to bring from this ugly one into the other ugly one. So, but this is, let's say, your target species, and what you do an F1, you do an F2, F3, and that is always backcross to them, and you reduce essentially the green background finally to 3% after only 5 backcrosses, and that is just showing the red part of that one. So after 5 backcrosses, you are almost back to your red mouse, which you had before, but you have now the gene of interest you have in there. Show you an example. Again, same story here, just to show you how it looks like on what the molecular geneticist actually is doing for us here. Is like this is a mutated mouse, because it's a 12 base pair deletion, so the segment will walk further in the gel. That is a wild type mouse. So you do your F1, and that is a F1, so you can see actually both alleles, and then you produce a litter of that one. And here you have, let's say, the mutated one, heterozygote. Here you have some homozygotes of that one. So you have all the segregation in this litter. Now you can make a choice of your selection. You do your marker-assisted introgression. I just show you that one. This is not just a trivial effect. This is not just significant, or these kind of things. This is like this mouse, homozygote carrier of that one, has a killing out percentage of 49%. And that one, the wild type, is 39. The fat percentage goes down from 12% down to 8%. So myostatin is also an anti-obesity gene, if you so want. And the muscle weight, some muscles were increased up to by 59%. So it's a drastic effect when you see that. So this was an example for marker-assisted introgression, which you can't do without the help of molecular genetics. Then it's another example here. It's marker-assisted introgression in chickens. They don't look very nice, but that was done in Israel, I think, the naked neck. So the layers which they imported from somewhere else, they identified the gene here, they introgressed it into here, and so they don't look nice, but they stand obviously, they eat much better. Polar overdominance, I don't want to show you all that one because of the time. I'll just show you the phenotypes of that one. Polar overdominance, it's again, there was a gene was identified, it's calipush, and you can certainly easily see that is, you would guess that is maybe the wild type. It's not that easy, but you can certainly see that they must be sort of carriers of that one. And I show you that was identified as a wild type and a carrier. And that is a carrier of that one. So it's heterozygote, all 3 of them with a very well developed hind-leg system are actually carriers of this calipush mutation, and they must have this copy from the father, otherwise they look out like that. So this animal could be everything. So it could be a wild type, homozygote wild type, it could be a homozygote carrier, it could have 2 mutations, or twice, a mutation twice on the mother and on the father's side. And still, then it is inactive, so it looks like that guy. And then if you get it from the mother, it's again, you look like that. And this phenomenon is called polar overdominance. Again, without molecular genetics, we would have probably still tried to work out what's going on here. Then this is Elisha Goodwine from Israel. He actually used this mutation, the calipush, and used marker-assisted introgression and put it into the Awasi sheep, which you might know better than me. I saw it first time as I was in Syria 4 years ago, so not recently. And so what he did was that in the night he took actually his sheep, has driven them to a hospital where they had a CT scanner, and he could actually show in a very nice way what CT scanning can do. He can actually see phenotypes these animals alive. So here's one thing to think about. If you find, of course, a gene you want to do marker-assisted introgression, and if you find a gene in a very undeveloped, let's say, breed, like you would find some good genes in a wild boar, then of course you do an F1, and then what you can see is, of course, you lose. That is like the progress which you make in your normal breed. You lose out a lot of selection response at the beginning, because that is like the wild pig would be here. You cross them, you have an F1. You lose quite a lot of all these generations which you put already in here, and then it takes a while, essentially a few generations, until you are at this level. And you need really to think if you want to use marker-assisted introgression. In summary, use markers to keep track of what you do while you are doing marker-assisted introgression. So you keep track of the heterozygotes during back crossing. Then you want to select the homozygotes after the same matings. However, also for speed up genome recovery. So you can just select for these markers which are associated with your gene, but you can at the same time also try to speed up that you get rid of all the other genotypic material which comes from the, let's say, undeveloped breed. You want to get rid of it as fast as you can, so you can also use markers for doing this. Some conclusions: the use of molecular genetics and selection program rests on the ability to determine the genotype for individuals for causal mutations, or indirect markers using DNA-based methods. This information can be used to assess the genetic value of the individuals captured in a molecular genetics score. If you so want now, it could be a genome-wide molecular genetics score. But highest efficiency only when the genetic architecture of the QT was completely transparent. We would know the number, position, and the effects of the involved genes. We would have a building block problem only. However, the black box of quantitative traits is yet only partially explained. So we learned quite a lot in the last years, but still there's a lot of other things. Other uses of marker, we're talking about marker-assisted selection or marker-assisted introgression. You can use markers also for product validation and traceability, for pedigree control and parentage confirmation. And if you are in the sheep world, you know a lot what's going on there, so you are really happy if you can actually really find out who the father was. Measuring diversity, an interesting one for conservation genetics. You can also predict hybrid vigor because you measure the genetic distance, and you can do it with a few markers, but the more the best is if you can do it, of course, with a SNP chip. And you can optimize the selection and control of inbreeding with using markers. And you can essentially do a selection on the one gene level. And you can also do genetic screening for major diseases. Some of them are just caused by one gene. I am not stupid to read it out. Then just a couple of words because my time comes to an end. On genome-wide selection you must be aware one could talk on its own just for 2 hours. But it was a very splendid idea of Theo Moiwiesen in 2001 that he was saying if we have the opportunity, the SNP chip was not out at the time as he came up with this idea, that we have not a few hundred, but we have thousands of markers all over the genome. Why can't we put a value, a genotypic value to each of these and put them all together and have actually a genome-wide breeding value? And that's what he developed, and then other people stepped to that one as well, like Mike Goddard and so on. And you have now, since that time, 2001, you have it now implemented in the dairy cattle in several countries. And the advantage of that one is, of course, that they are all working with the same breed. So they can actually swap genotypes, forward, backward, if some countries decide to work together. Then it's because you must know that you need essentially several thousand animals to do genome-wide selection in your trainings population. And so types of selection, just to wrap up here. So that is the normal what we are doing at the moment. We have our phenotypes, we have the pedigrees, we use blood, and that works fine except for the difficult to measure traits. And for example, maternal traits are difficult, longevity is difficult because mostly animals are gone before they have actually seen the end of their life. Then carcass traits and meat quality especially. We are fighting now for some methods that you can measure meat quality online, but the problem is still that you need also traceability aside of that one, that you get this information back as phenotypic value. For disease resistance it's also difficult. Then if you have individual markers, QTLs like the TMQTL, Myomax or other ones, the problem is there are some good examples, but most traits are controlled by variation in many genes. And then you can use this genomic selection, and for example, many use the 50K SNP chip. Now, some of them have already used on several hundred of these animals 700K SNP chip, but that is mostly on cattle. In Australia, they have Australia and New Zealand, they have progressed also using the 50K SNP chip on sheep. And the question is really what are the costs? The cost went down now. I think you pay $100 probably for one animal. Then the questions are low density and high density SNP chips. For the best strategy, you actually want to have a mixture of low density SNP chips because they are cheaper, and you want some high density, and you use an imputation to essentially reconstruct, the information which is on the high, but having only the information from the low density one. And then it's of course, now we realize that the SNP chip is very breed specific. It's a further way to go. And even the 700K SNP chip is not working very well across breeds. And though people are discussing also the SNP chip design here, so should we have more SNPs which are associated or in the neighborhood of genes? And for that one we need to identify these regions and so on. So genome-wide selection I have just touched on. And genome-wide selection potential benefits would be that you can, of course, reduce the generation interval enormously, especially in cattle. I mean, normally a bull needs 5 years before he can actually say he's progeny tested, and that can go. We have a higher accuracy, we have greater progress to be made in difficult to measure traits especially, and we have evaluation of greater numbers of selection candidates can be done. The cost of progeny testing could be reduced. Possibly. And yeah, I just threw that one up to say that the next step in the development will be sequencing. And then we have the big task how to digest all this information and what kind of computers will we need if we have for all selection candidates, this kind of information available. But it's already useful when we have highs and lows, when we can actually compare different groups, disease groups, non-disease groups, and all the rest of it to identify more and more genes. So this will be the thing of the future, and as I said, 10-fold or maybe even 20-fold coverage of the genome for one animal. Probably it will come very, very close to $1,000 in the next time. I'm not quite sure if it is already the time for it.
No,
probably
no.
not.